All right, welcome and thank you everybody for joining our Sargassum webinar series. We're launching this series to raise awareness about key issues surrounding sargassum. Each webinar in our series will highlight different aspects of sargassum research, management, and its impacts on coastal communities and ecosystems, as pictured here on this graphic. The goal of this series is to foster knowledge sharing and regional collaboration across affected areas as we continue to see impacts from sargassum inundation events. My name is Catherine Egan. I'm the Southeast and Caribbean Regional Coordinator for NOAA based in Key West, Florida, and one of the hosts of this webinar series. I help co-lead and coordinate for NOAA's Southeast and Caribbean Regional Collaboration Team, also known as SECART. SECART is one of 8 regional collaboration teams across the country, working with both NOAA staff and partners throughout the United States to foster stronger collaborative ties in an effort to improve NOAA's regional capabilities and services. You can find more information on SECART by following the link that'll be put in the chat in just a moment. So here are the webinars that are taking place during our series. Today is our second webinar out of 5. You can scan the QR code at the top of the slide, and that will take you to a website where you can view descriptions of all of the webinars and also register for the remaining 3. I'll also put a link in the chat as well if the QR code is not working for you. All right, so with that, I would like to introduce my co-hosts today. With me are Max Brown and Josie Galloway with NOAA's National Centers for Coastal Ocean Science, or NCOS. So I'll pass it off to Max to introduce himself first.
Thanks. I'm a contractor working with NCOS as a program analyst, so I have a background in coastal ecology, phytoplankton, and some molecular biology. I started working on sargassum management last year, where I helped pull together an assessment of federal policy. And following that, I sort of dove right into helping organize a workshop identifying research gaps. Pass it on to Josie.
Thanks, Max. Hi, everyone. I'm Josie Galloway. I work with Max at NCOS. My main role is that I'm the Executive Secretary for the Interagency Working Group on Harmful Algal Blooms and Hypoxia Research and Control Act. This group sits under the National Science and Technology Council. In that role, I help to coordinate with federal agencies to develop strategies to research, prevent, control, and mitigate the impacts of HABs, and this includes sargassum. Over the past year, I've helped Catherine and Max with the policy assessment and the webinar series and the workshop. Before I pass it along to Catherine to introduce our speaker, I'm going to go over a few housekeeping items. First, all participants will be muted during the presentation, but you can send us your questions in the question box and we will answer them at the end. Second, due to a large number of participants, some of the slides might be delayed as we pass through them. And finally, we will be recording this webinar and we will put the link on our website so you can watch it if you're unable to attend. With that, I'll pass it to Catherine to introduce our speaker.
Thanks, Max. Thanks, Josie. All right, so the title of our webinar today is Tracking the Drift and Origin of the Great Atlantic Sargassum Belt. I'm pleased to welcome Dr. M. Josefina Olascoaga, who's a professor at the University of Miami, Rosenthal School of Marine, Atmospheric, and Earth Science in the Department of Ocean Sciences. Josefina, I'm going to go ahead and move screen sharing permissions over to you so you can get set up. And while she's getting set up, I'll go ahead and introduce her to the audience today. So Professor Maria Josefina Olascoaga, specializing in ocean sciences at the University of Miami, uses nonlinear dynamical systems theory, including Lagrangian coherent structures and probability theory, to investigate the effects of ocean currents on biogeochemical processes. Her research includes predicting oil slick dynamics, exploring harmful algal blooms, examining larval dispersal, and studying sargassum movement and distribution. Originally from Buenos Aires, Argentina, she received her degree in oceanography from the Instituto Tecnológico Buenos Aires and went on to obtain a master's and doctorate in physical oceanography from Centro de Investigación Científica y de Educación Superior de Ensenada in Mexico. She did her postdoctoral research at Erasmus and became part of their faculty in 2008. So Josefina, welcome. We can see your presentation, but not in full screen mode. There we go. Now we can see
Okay.
it. I'll go ahead and pass it off to you to begin.
Okay, thank you. Can you hear me?
Yes, we can hear you well.
Perfect. Um, so today I will present some work done in collaboration with Javier Berombera, Gage Banner, and Philippe Miron, mainly, and others as well. This started as a pilot project with NOAA, and we received funding from NSF to continue this study. So this is the second series of Sargassum, of these webinars. And what we are trying to talk today is basically where it came from. So I will first get back a little to what was um, presented last week, but very briefly. But the main focus of the talk today will be about how we model sargassum and how we can use that modeling for finding the origin of the Great Atlantic Sargassum Belt. So mainly I will present some of the hypotheses and what are the gaps that we still have. So before going to the historical distribution, I just want to emphasize a couple of things that will be important for sargassum. That we not only have a clamp that is few centimeters, but they form mats that can be very large, and they are buoyant, so and they can expand deeper in the water column. So it's not just a few centimeters; it can go to 2 meters below the water. So this will be important to think about how it's affected by currents and wind. So the historical distribution is as presented last week, mainly on the Sargasso Sea, on the subtropical gyre, but also on the Texas shelf. And this is we have evidence of sargassum in these two areas from very early. So we have reference from the explorers and newspapers in the region of Texas as early as 1900. So, but what is new is that this sargassum start blooming on the tropical Atlantic. That was evident, or the first big event was in 2011. And once in summer, the bloom can cover the whole basin, going from Africa all the way to America and enter the Caribbean, and affecting, as is well known, all the Caribbean region and this east of South Florida. Sorry. So before going to the hypothesis, I just want to briefly mention the main currents that we have. In terms of the subtropical, we have the subtropical gyre that um, where the Sargasso Sea is bounded, and the new bloom is in the tropical region where we have the equatorial current system, mainly going from east to west, but we have a current, which is the north-equatorial countercurrent, will go from west to east in the basin. So this north-equatorial countercurrent is important to know that it has a seasonality. So what I want to point out is that the currents in the equatorial region has a pattern that allow things to stay there for a while. So the hypothesis that Jones et al. In 2020 brought says that the origin of the sargasso that we have in the tropical Atlantic is coming from the Sargasso Sea, and it's mainly this leakage was mainly done because of a very strong now event in 2009, 2010 that brought the sargassum all the way to the east and then moved to the south through the Canarias Current and enter the tropical region. In a really recent study, Junano et al. Agreed with that, this finding, and it's mainly, I'm sorry, I didn't put this, it's a modeling study where they started the sargassum initial condition in 2003, and they can show an intrusion that culminates in the bloom of 2011 and so on. I want to point out that there were other studies previously, like Brooks et al. In 2018, where they found that the Sargasso Sea was only connected with the tropical Atlantic as a sink source, so the other way around, nothing coming from the Sargasso Sea to the tropical Atlantic, but from the tropical Atlantic to the Sargasso Sea. So from this main hypothesis that the Sargasso Sea is the origin for the tropical Atlantic bloom, we have some gaps and maybe some overlooked observations that I want to point out to continue and see what we can do next. Okay, so the first thing I want to point out is the species composition between the Sargasso Sea and the tropical Atlantic is different. And this was, there are a couple of studies that show that. I mean, I didn't mention at the beginning, but the pelagic sargassum, we have two species, fluitans and natans. The Sargasso Sea is mainly dominated by natans 1, where this is a study by Parr in 1939. And in the only region that he found Nathan A was in the southern part of the Caribbean, and pointed out that this probably was coming from the tropical Atlantic. More recent studies also shows a similar distribution, observations in 2014 and observation between 2015 and 18, where the tropical Atlantic still has mostly a Nathan 8 and Nathan 1 is in the Sargasso Sea, as well as flutans. So that brings a problem, right? So if the source of sargassum is the Sargasso Sea, why the species are different between the two blooms. Another thing is a report that the first landing of sargassum in the tropical Atlantic was reported in the Gulf of Guinea in 2009, and that is actually too early to be transported from the Sargasso Sea after the NOAA event. So this is another thing that we would like to evaluate further. One thing that I think was also overlooked is that there is a pre-existing baseline concentration in the tropical Atlantic. So from satellite, we have observation by Gower et al. In 2013, where still small, but you can see yearly peaks of sargassum in the tropical Atlantic. Of course, they were nothing as the ones are recorded after 2011, but there is presence of sargassum in the tropical Atlantic before 2011. We also found many papers and descriptions of species that have record of sargassum, especially in Nathan's 8, prior to 2011 in the basin. So all these things are points that questions if we really have the tropical Atlantic bloom origin in the Sargasso Sea. This brings us to rethink the origin of this Great Atlantic Sargassum Belt, and we have some observations and modeling that I would like to present today. So first of all, I will present the first part of the talk. I will present a model where we want to incorporate buoyancy, size, and elasticity for transport the sargassum. the second part of the talk, I want to use that model to find the origin for the tropical Atlantic bloom. Okay, so the way we will do the modeling, as I said, we will consider it that is floating, but it's buoyant. So part of the sargassum has exposed to the atmosphere and part to the currents. It's not, as I said before, isolated, but forms big mat raft. So all those things are not bring other forces that are not the same as modeling just a water particle. So I have just this slide with the equations, and I will try to go quickly. But what we did in the paper of 2019, we follow Maxi Relay um, for a similar problem where we have a sphere that is exposed to the air and also to the currents, and what are the forces that that object will have, right? So we have a drag force, we have the flow pressure in the object. We also have, when the object is moving, is carrying a mass with it, and so on. So if you put all these equations in a Newton's kind law, what you will obtain is that the trajectory for the Sargasso, so x dot, is equal to a velocity, which is leeway-like, is a combination of the current plus wind with the parameter here, which is the windage alpha. But we have an extra term here, which depends on the size and other forces that I usually smaller, but it's important for certain parts, like entrainment into eddies, cyclonics and anticyclonics. Okay, the important thing from this is that we usually, or previous modelers, what they do usually is move the sargassum with a leeway kind of law, where alpha is usually used between 1% to 2%. What we have here following the equation is that we can find alpha, or so the windage, in closed form as a function of the buoyancy. So how deep the sargassum is, or what's the volume above and below the water, will tell us which is the windage that we need to use. The next thing that we need to take into account, and this is a work by Javier and Philippe in 2020, is that we don't have isolated object, but we have connected, interacting aggregation between them, right? So the way they model the sargassum is as using a hook law, where you have the springs within these buoyant particles. Okay, so using this model, the first thing that we did, we test part of it in the field and in the lab, and we used that experiments to obtain some of the parameters that we didn't know. So the first experiment was done in the wind wave tank at Rasmus. We actually didn't have waves. We just used 40 realizations of different winds and currents, different combinations. This was published in 2023. And what we did, we measured the velocity of a sargassum. So we measured the distance, the time, we obtained how fast the sargassum was moving. We knew, because if we are in a control tank, we knew the current and the wind under that the sargassum was moving. So from this equation, we can actually obtain what was the windage that sargassum was feeling. And because from theory, we have a closed form from the windage, we could obtain as well the buoyancy of that sargassum. So the results are as follow. We found that the windage was actually much smaller than the usual values of 1%, so it was 0.5%. And to check this, what we did, we invert and obtained the buoyancy and compared with those values that we obtained from direct measurement of the buoyancy in the lab, and they have a good agreement with that. Okay, so we have now a model that is depart from the traditional leeway modeling from especially because we can have closed form from some of the parameters, and we will use this first to I'm showing here an example from a paper of last year where we start initial condition for March 2018, and in the figure we have satellite a month later and 2 months a week later, and in the second column we have a line, we have results from the model, and in the third we have results from the leeway with 1%. So first of all, the comparison it looks well, but we will have a quantification just in a couple of slides. So the next step was, for now, is all physics. So what we did in that paper as well, we had a very simple life cycle. Basically, for the modeling point of view, depending on if the sargassum is growing, we will add one more equation, or if the sargassum is dying, we will eliminate one equation, right? And the way that the sargassum is growing or dying, we have following Brooks et al. We have a growth rate that depends on light, temperature, and nutrients. And for now, a constant mortality. That was what we had in that paper. Probably this could be a function of temperature as well. And of course, this model can be as complicated as you want, right? So this is a first attempt to add some biology with the physical model. Okay, so here we have the error from the images that I presented in the previous slide. So we have the comparison. Sorry, this is the error is measured. This is actually measuring similarity between two probability distributions. So in blue we have the physical model. In red is when we add the biology, that it has a little improvement, but in comparison, both errors are actually much better than if we model the sargassum as leeway with 3% of windage or 1% of windage. I want to point out that well, this model is already published, and we put available in Julia with an interface. So you can actually run your own experiments, changing parameters and everything from here. So just a quick summary. We develop a physical model that incorporates buoyancy, size, and the raft structure. The comparison with the satellite of observations under currents and wind from reanalysis. I think I didn't mention that, but we didn't run a model, but we used the currents and wind from reanalysis. It has a good comparison. Some of the parameters were obtained, as I said, for example, the windage from the wave time experiment. We are considering some biological processes and more maybe here to do. And this is available in Julia. So now that we have the tool, what we want to do is actually some work that is under review right now. We want to use this model to infer the origin of the Great Atlantic Sargassum Belt. So we focus first on the first bloom that was evident before the big bloom in 2011 was in April 2011, sorry, before the one that covered the whole basin that was more on the summer of 2011. So the first bloom that we see from satellite was in April of 2011 offshore Brazil. And so what we will try to do is use actually three different ways to infer the origin that are independent. Okay, so we want to get the focus will be not in individual trajectories, but more on the probabilistic framework. So what we will do, we will um, build this transition matrix. What is this? I should mention this is a timing homogeneous, but I will just explain it at a fixed time. So we have all the domain divided in boxes, and basically what we do, we start some trajectories and count how many from this box end up on this box or in this box, and so on. So we build this transition matrix, but instead of having just one transition matrix, we will have them as a function of time. Okay, so once we have the transition matrix built, we can use this to, for example, push forward. I want to show how the transition matrix works. So if we start with the probability of the bloom in April 2011, and we push with several p's here, not just one, and we advance in time just by multiplying by the transition matrix. By the summer of 2011, we have the bloom covering the whole tropical Atlantic basin. Okay, so now we will use this transition matrix for the first methodology that we will do for will be to find the origin. In this case, would be Bayesian inference. And what we want to do here is we want to find the likelihood location of the origin that will end up on the bloom position, capital B, the blue, in April 2011. One difference with previous studies that we did using this same methodology is that in this case, we don't know actually the day or the time this bloom the bloom originates, right? So we don't have one unknown, which is the location of the origin of the bloom, but also we don't know what is the time of this origin. So what we did, we expand and redo the analysis within a period of time that cover times that we thought were logical from previous studies and from the knowledge or observations like the ones I was describing a couple of minutes ago. So basically we start this in 2009 because it's when the 2009-2010 is the now extreme event. So for comparison with the Sargassum is from the origin of the as the Sargasso Sea. And we also want to include the 2009 because we know there are reports for a bloom in the Gulf of Guinea during 2009. So the results are as follows. So if we go just one year back, so in April 2010, what we have in colors are the probabilities. So you can see that you can see some gray colors and white colors in most of the subtropical Atlantic, but the darker part are within the tropical Atlantic, with the maximum in the coastal area of Africa. If we go farther and we go to, of course, there are intermediate times, but I'm just showing 2 times. And if we go farther and we go to the beginning of 2009, what we see is that the maximum probability is around the same place of Africa and including the Gulf of Guinea. So this is the first analysis that I want to present. The second one is what is called transition path theory. And in this case, we will select 2 areas, A as the source and B the sink area. So A from where it's coming from to where it's getting, and we will find the more productive currents. So we are looking for things that are not getting back to the same place or give a lot of loops before getting from A to B. So the first thing, what we present here is we selected as origin 2 possibilities: the Sargasso Sea or the area we found from the previous analysis. So I want to call the attention to the bars here that are, even though we have probability for coming from the Sargasso Sea, if you take a look, it's 2 orders of magnitude smaller than the currents that are coming from the west, from West Africa. Sorry, I didn't mention, but again, we start, the initial condition in A was in January 2009, and arriving or final time will be in the region offshore Brazil in April 2011. Okay, so the next step, and it's the third thing we try, and on the last one. So we say, okay, we don't want to say from where it's coming from. So we are actually saying that B is the target and is in April 2011, but instead of saying it's coming from the Sargasso Sea or from the west coast of Africa, what we will say is that the complement of B anywhere in the domain can be the origin. So the results are there for several times. And what you can see is that most of the effective currents are within the tropical Atlantic region. Okay, so all the things that I presented until now for the origin were exclusively based on physical constraints. So we didn't check anything related to temperature, salinity, nutrients, and that is something that we need to consider. So we consider that there was a pre-existing baseline concentration of the sargassum in the region. And physically, using the model that with the physical constraint, we found the origin most probably in West Africa, as I mentioned. But if were always some sargassum in the region, there could have been blooms like the ones we got in 2011 at any time, right? So one of the things we want to check, or we want to know, what was exceptional or what was different in 2011, if a bloom can be created just starting from the baseline concentration of sargassum in the tropical Atlantic. So we check the environmental conditions within the region. Within the region that we have the maximum likelihood and what we found is that these are all data from reanalysis. So what we found is that in 2009, there was the sea surface temperature was much lower, and the nutrients actually has a peak during that same year. When we look for the optical depth to have an idea of what was the amount of Sahara dust, what we found also is that it was an increase during the 2010. Let me just so these were the things that we found using biogeochemical reanalysis, but we found some papers that corroborate or give us some idea that something is going on within those places at that time. So in this paper, they introduced what is called the Dakar Niña Niño. And what I am showing here are the index for Dakar Niña Niño that are actually in the area of Africa that the latitudes and longitudes are there. But you can see here that the wind anomalies and the temperature anomalies. So the wind is a the positive anomaly, and the temperature is a negative anomaly. And you can see that this Nina Dakar event was actually really strong in 2009. So what is a Dakar Niña Niño is a similar to La Niña Niño event that we have in the Pacific or the Atlantic, but this is a coastal mode, which has a coupling, ocean, land, and atmosphere. And of course, it is related as well as the tropical conversion zone is moving, and also the North Atlantic Oscillation. So all these modes are related, but the signal is there in the area where we found the maximum likelihood for the origin of the bloom. And similar modes that this Dakar Niña Niño, coastal Niños, are found in other basins in the eastern boundaries. And the other thing that I want to point out are not from satellite, but in situ observations of the Sahara dust. So I want to point out two papers. I know some people, the hypothesis of the Sahara dust was related to the initiation of the bloom was mentioned many times, but and I saw just one work, but mostly on the Caribbean, not on the region of origin. So I think this deserve a little more of a study. So these are papers that are not related to sargassum, but what I want to point out is that from in situ observation of Sahara dust in Barbados, the dust is actually decreasing, but the amount of nutrients is increasing. And the other thing that I want to point out is that the timing of the arrival is also changing. So that are things, this is done by season, so winter, spring, summer, and fall. And you can see that the anomaly is positive within the years of our interest, mostly on spring and also in summer. So there are more things to check in relation to sargassum and this data, and this is work that one of the students here is starting. So in summary, I just want to point out that we did 3 different ways to infer the origin, and all of them point out to the west coast of Africa. This is consistent with the report of 2009 landing in Ghana. For Additionally, we have unusual environmental conditions that include the strong Dakarninia and pronounced that's the position within the region as well. So our findings challenge the view that the origin is in the Sargasso Sea, and mainly I will think in terms of the species. So if the original bloom of 2011 was mostly Nathan's 8, and the pre-existing amount of sargassum in the area baseline was also Nathan's 8, is more the hypothesis that the origin is actually within the region has more support in that sense. Given that in the Sargasso Sea, the composition is different and the main species will be Nathan 1. And with that, I think I can finish here. And I want to mention as well that the related publications can be found in the webpage. From these two projects, there were several publications, but the ones in yellow are the ones that I focus or I use today for the results that I presented. Okay.
Thank you so much, Josefina. That was a fantastic presentation. Folks, feel free to utilize the question box in the upper right-hand corner if you have any questions for Josefina. We did have a few that came in. I'm happy to start off and then I can pass it off to Max to tackle another question. This one came earlier in your presentation, and this person asks, can you elaborate more on why the species composition of sargassum is different in the tropical Atlantic versus the Sargasso Sea?
Well, I mean, it's not that I am saying, right? These are from observations. So is there? Can you see the figure?
Yes, I don't know if you want to make it full screen, though.
Yeah, sorry. Yes. So there are observations before, historical observations before the bloom of 2011 and at the beginning of this new era, if we want to call it, with the tropical Atlantic as well as the bloom. And the observations are that in the tropical Atlantic, the main species is Nathan's 8 in comparison to Nathan 1 or Fluitans in the Sargasso Sea.
Thanks, Josefina. Max, do you want to tackle
Yeah,
the next question?
yeah, definitely. I think the next question in the queue would be asking actually, given that the Deepwater Horizon spill, I guess happened in a similar time frame to the first appearance of sargassum, asking if you could hypothesize, I guess, for any possible connections there in the Mid-Atlantic.
No, I don't think so. And also, I want to point out that the Deepwater Horizon was in 2010 and the first landing was in 2009. So it's also.
Can I follow up there actually with my own question about how fast does sargassum move? Like, what's the speed approximately in the water? Like, what are you using? I guess it varies with the currents, right? But like
Yeah.
ballpark, you know?
So of course we'll change with the currents, as you are saying, and there is a contribution of the wind, and so depends on where you are, uh, will be the velocity of the sargassum.
I guess what's the maximum sargassum velocity?
Well, I don't know. If you are in the Gulf Stream, you will go 1 meter per second plus the winds that are pushing you toward the coast. But if you are, I don't know, somewhere in the middle of, I don't know, where there are no strong currents, you may be much slower than that.
Yeah, I'll pass it to Josie to pick out the next question.
Thanks, Max. So, one of the questions we got was, what is the best place to track real-time sargassum, such as monitoring the size of the destination? And I'm actually going to put the Caracoos tracker, sargassum tracker, in the chat here for anyone else who wants to see it.
Yeah, that's
Yeah,
that's that's actually very good.
perfect.
So yeah.
All right, then we can go to the next question.
Sure. Yeah, I'm happy to tackle that one. So this next question, this person asks, based upon these analyses, are there areas of the Great Sargassum Belt that we might expect to see larger blooms of sargassum in the future, independent of transport? And what do you feel is the driving mechanism behind the dramatic increase of sargassum in 2025?
So a couple of things. So because of, as I said at the beginning of my presentation, because of how the circulation is in the equatorial region, you will have things that keep going back and forward, and some, of course, will end up going into the Caribbean. And of course, it needs some source of nutrients that will maintain that bloom. So, not from my own research, but the vertical mixing is what most people are pointed out for growing or maintaining the Great Sargassum Belt in the tropical Atlantic. So
All right, thank you.
one thing I didn't mention, and I didn't mention because I don't think it's related, but another hypothesis, now that I mentioned the nutrients to maintain, was related to nutrients from rivers. And I didn't do any specific because there were there were more than few papers talking about the subject before our work, and there is nothing specific for the 2009-10 time that could be related to the big rivers on the basin.
Thank you so much. I'll go ahead and pass it back to Max for the next question.
All right, thank you. Questions are very good so far. People are really asking from all angles. This one is, with the amount of export from the West African region, do you think there is a persistent local population that is serving as a source?
Well, yeah, so that is coming from previous documents, previous to 2011. So it was already a small amount of sargassum in the region.
Awesome. All right, Josie?
Yep, more questions about the sargassum species. And I hope you know the words that I'm about to say because I don't know exactly what they mean. But regarding the S. natans morphotypes, are they phenotypically plastic ecotypes?
I will have to say that I am not from biology. No, I don't know what.
All right, so
I
we might need
don't.
some
I
more
mean,
more help for that.
I will have to refer to some of my colleagues that were probably on the. Yeah, no, no, I can. I
I
don't want to
think
say
I
something
think
wrong.
I think the
Sorry,
question is asking if they change their appearance depending on where they are.
they change the appearance.
Yeah, phenotypic plasticity, meaning they look
Okay,
different in different
sorry.
situations.
No, I cannot answer. I don't know. I mean, they look different, but and I and probably they have different characteristics. That, but I no, I cannot answer that.
All right. Um, yep, well, we need a biologist to chime in
Yeah.
the questions box. We have our physical oceanographer on the line. So, all right, well, thanks, Josefina. We do have a couple more questions that are coming in. So I'm happy to tackle this next one. This one, it's a little vague, but this person is asking, can you expand on the sargassum model showing the rate of change with time?
Showing
Showing the rate of change with time. I
I'm not
think maybe this person didn't complete their question, so if you are out there
okay.
and you ask this, please follow up with us. Um, I'm
Yeah,
happy to jump to the next question though.
okay.
Okay. We'll go ahead and skip to the next one. Okay, so this person asks, could there be two source regions bringing different sargassum species, but then one sargassum species wins out?
Yeah, of course. Yeah, and that's something I hope it was clear within my presentation. I'm not saying that was impossible, that sargassum was actually getting to the tropical Atlantic from the Sargasso Sea. I am not saying that, but I am saying is that um, there was already sargassum in a small amount in the tropical Atlantic, and it was the species that was actually the big bloom after. So in 2011. So and of course the story changed a lot because today, and I mean, blooms lately are not mainly Nathan 8. So now that we have the region in permanent bloom, let's say, there are other species are also, yeah, in the inundations you do get fleetons and Nathan 1 as well, right? Mostly fleetons in the Caribbean.
Great, thank you. Back to you, Max, for the next question.
Awesome. I like this question. It's about the nutrient data that you showed. It's asking what is the exact location that those nutrients were
Oh, yeah.
sampled.
Yeah, I realized when I was talking that I didn't have any. Let me get to. Um, sorry. Oh, okay. So I guess this map will do it. So the region where we are showing the environmental conditions are within this maximum likelihood. So the coast of Africa getting into the Gulf of Guinea. And I hope that clarifies.
Yeah, I think so. And I
So,
think
and
they were, I think they were trying to understand how their question says, could you expand where the nutrient data are from? I'm
yeah,
trying to
so
understand
yes,
how.
of course I can. So as I mentioned before, so these are average values. So these are getting all the data for that specific year in the region I just mentioned, and the data is from a reanalysis. So it's a BioGlories, I think it's called, um, and is average in time and space within that region.
All right, great, thank you. Moving along.
All right, the next question that we have for you, can you address how mat thickness might impact the models?
Sorry, how what?
Can you address how mat thickness, like how thick a sargassum
Ah,
mat
yeah.
is, may affect the
So,
models?
yeah. So actually, um, that is something that we are taking into account with the buoyancy. So, but of course, it can change a lot. It can be much thicker than, but the important part here is that the volume that we have above the water is much smaller than the volume we have below water. But in terms of size and volume to be transported, that it may have yeah, some effect that we probably didn't consider.
Thank you.
Yeah, thanks, Josefina. We have a lot of questions coming in. This is great. Thank you all so much. We're going to go ahead and address a couple more. We have like 5 minutes left today. Our next question is, what oceanic and atmospheric conditions favor the growth of sargassum at its point of origin? And how does climate change affect the frequency and scale of sargassum blooms?
So the atmospheric conditions in the region, the map is there. Sorry, the time series are there. So in the region of origin, you have it's mostly an Apollin region, right? And in this particular year, there was extra amount, or the anomaly was much bigger because of this coastal event. And you also have, as we were seeing from the data, some extra or some anomaly of dust as well. And the other thing that I think is important, but we haven't done for the whole basin, is this timing in change, the change in time that the dust is arriving and the composition of the dust. So that's probably something that needs further investigation.
Thank you. Uh, Max, next one.
Thanks. There's a couple different questions I think that are about modeling and basically forecasting years. So I guess I'm going to try to lump them into one, which is basically saying now that we've for the 2011 event, we've sort of done the work to estimate where and how, what the mechanism was. Can we, using satellite data, maybe nutrient data from the similar area, can we then project out, like, into the future, like what years are going to be bad and good?
Yeah, so the tools are there, and there are other authors that as well presented different models. So there is a prediction tool, if you want. The one that I presented here, as well as others, as I said, may have something a little different. But yes, and the other thing that the people are sometimes reporting is based on just a satellite and comparing with previous years. So depending on how it looked like today in this region and in compare. But I mean, that's not actually a prediction, but they also have some inference from um, how it may look like in another place.
Do we have time for maybe one more quick one?
Yeah, I'd say one more.
All right,
Great.
Josie.
Good. This is a good one, I think. So, is there a reason for such a large research gap in studying sargassum movements? Was there not a lot of interest between the 1930s research from Parr and the early 2000s work that focused specifically on the movement of sargassum?
Um, Yeah, probably because it was not a big problem, right? But there were, not in terms of transport, but in terms of the biology, yeah, there is a lot of work done. So, I mean, it was not the focus of my talk, but in the 80s, you have a lot of work specifically on nutrients and temperature with sargassum. um, I want to say the 80s, and I will probably, I don't want to mention, there is work by Lapointe, but there are many others that work on that at that time, but not as much about transport. I don't think I saw any paper, but that time, I think it all started. Probably the earliest modeling are the ones by Brooks group, Victoria Coles as well. Mhm.
All right, great. Yeah, like I said, we got a lot of questions today. I wish we could get to them all, but we do need to wrap up. I put um, Josefina's email is up here on the screen. I also have our generic Southeastern Caribbean email here in the chat. Feel free to reach out if you want to follow up on anything we heard today. But just want to say thank you so much, Josefina, for the really wonderful presentation today.
Thank
We
you.
really appreciate you being here.
Thank
And folks, next week we have our third webinar in the series. The link to register for that is now in the chat. That's going to be on July 24th, next Thursday at 1 p.m. Eastern time. If you can't make it, we will record it and put it up on our Sargassum website right here at a later date. So with that, we'll go ahead and conclude the webinar. Thanks again, Josefina. Appreciate it.
Thank you. Bye-bye.